because the more of the drug is having affinity, the drug has more affinity towards the particular tissue, because of which the more drugs start getting accumulated in the tissue, and because of that reason the volume of distribution, when you calculate the volume of distribution, the value will be much higher. Why that value becomes higher? Because the plasma concentration, this rule you remember, when the plasma concentration is at lower side, definitely the volume of distribution will be higher side. So lesser the plasma concentration, larger will be the volume of distribution. And lesser the plasma concentration can happen because of many reasons. The one reason is it can be because the drug would have gone and attached to the tissues because of any of these reasons, or the drug would have got metabolized. It would have not gone and bonded to the tissue proteins or the lipid components, or not transported across the enzyme system, but it would have got metabolized. Even the drug getting metabolized means the normal form of the drug, and that is the normal chemical structure of the drug would have changed. So whatever we measure the concentration, it is the original structure, that the original drug molecule. So we are not actually measuring the metabolite. So even in that case, because of the metabolism, because of the affinity of the drug towards the tissue for any of this reason, there is always a chance that the volume of distribution will be much higher than the normal value. Okay. So the digoxin and example, we got an idea why it is showing the high volume of distribution, and difference in the adult and children is it's because of the body mass or the age difference, age conditions, because infants, children, adults, there are a lot of changes with respect to the body components, or the that is the volume of the tissue will be different from the adult to child also. So next one is the effect of the protein. What happens to the volume of distribution? I already revised it before also. Drugs start binding to the protein, and this protein can be, the one is the plasma protein, another one is the tissue protein. Okay, so the extent of drug protein binding in the plasma or tissue. When I say drug binding to the protein, don't just imagine the plasma protein. Previous slide I just told you, even the cellular protein. Cellular protein is outside the systemic circulation. So the protein binding can happen even at the plasma level, even at the tissue level. So if the drug is bounded to plasma, I told you, because it binds to the plasma, the molecular size of the complex increases, because of which the drug cannot easily cross even the capillary membrane. Why I say even the capillary membrane? The capillary membrane is one of the permeable membrane compared to one of the high permeable membrane compared to any other membrane in our body. Okay, even the normal, that is plasma membrane we talk about, compared to that also the capillary membrane is highly permeable. So that's what we already discussed in the beginning of the distribution chapter. It is generally highly permeable to most of the drug molecule, exceptional in the blood-brain barrier, blood-brain cerebrospinal fluid barrier, placental and testicular barrier. In all that cases, because of the change in the structural arrangement, the cellular arrangement, there is a chance that they are slightly impermeable to most of the drug, and that depends on the hydrophilic and lipophilic property of the drug molecule. So, okay, all that. So, protein binding drug, even in a normal, it cannot easily cross the normal capillary membrane also, because of the increase in the complex structure, that is size of the complex, and because of which what happens, the drug cannot cross the blood. That means it is still present in the plasma. When I measure the plasma for the concentration of the drug, I can measure even the drug bonded to the plasma. So it's analytical technique we have. So what happens? Definitely the plasma concentration value will be high. When the plasma concentration value is high, then definitely the volume of distribution becomes low. With respect to this previous statement, we got to know that lower the plasma concentration, higher the VD. Higher the plasma concentration, lower the VD value. And this is the relation I'm just giving you to get idea. So protein blood drugs results in the decreased volume of distribution. So low plasma, vice versa is also true. Low plasma protein binding have greater free drug. This is justification. Why that unbound protein drug has larger VD? Because the free drug presence is more, and this free drug can easily cross the capillary membrane, and because of which it can easily reach the tissue, and that results in the greater volume of distribution. So protein binding effect with respect to plasma protein, we got to know that Protein bonded drug, more the complex structure, the size of the complex structure, and because of the greater size, it cannot cross the capillary membrane, because of which VD will decrease. This is some of the examples to add up to that. Furosemide, sulfasoxazole, and torbutamide, all these drugs and warfarinols are bonded greater than 90% to the plasma proteins. Okay, and because of which they have a VD value, that is a volume of distribution value ranging from 7.7 to 11. So these values I don't expect you to buy it also, but look at the values, the number. It is 7.7 and 11.2, which is much less, like 40, even doesn't come closer to the 42 liters. At least if it comes to the 42 liters, we can say it is distributed to the tissue. Okay, so this drugs, because of the higher protein binding capacity in the plasma, its VD value is decreased with respect to 70 kg body weight. Okay, so basic drugs, these are the other examples of imipramine and nortriptyline and propranolol are extremely bound to both the tissue and the plasma protein, and this is some exceptional cases. It is bonded even to the plasma protein, and also it has more affinity towards the tissue protein. So there is a chance that in this case there is a possibility of larger VD values, because maybe its affinity matters now, whether the drug has more affinity to the plasma protein or it has more affinity towards the tissue protein. Let us imagine a drug A which can bind to plasma protein as well as it can bind to the tissue protein. If the affinity of that drug A is more towards the tissue protein, then it binds more to the tissue protein than the plasma, because of which VD value will increase. Means this is just an example for you that though the drug has the binding capacity for both the plasma protein and the tissue protein, depending on its affinity, there is a chance that VD might increase or VD might decrease. If its affinity is more towards the plasma protein, then VD will decrease. If it has more affinity towards the tissue protein, then the VD will decrease. If the protein of plasma, it is having more affinity, it decreases. If the affinity towards tissue protein is more than the VD increases. That's what's written in the bracket. Favorable tissue partition coefficient and the protein binding in tissue. That depends on the physicochemical property of the drug molecule. So when we talk all this, we should also know what's going to happen to the elimination of the drug because of can we relate the volume of distribution to the elimination half-life of the drug? Let's see. So extensive drug distribution has effect of diluting the drug in the large volume. Acceptable. Making it harder to kidney to filter the drug by glomerular filtration. See, if the drug has more affinity towards the tissue, it's holding. Okay, imagine it's holding, just holding. For the drug to get eliminated out of that, it should be released from the tissue. It enters back to the systemic circulation, and that blood flow towards the kidney, and there the glomerular filtration might happen. Okay, this is what actually the sequence. Okay, thus the T of the drug is prolonged. Why the T half of the drug is prolonged? The tissue is not leaving the drug. So extensive drug distribution, it is so much of distribution would have happened because of what reason? What all reasons we have just revised? Okay, whatever the reason may be, but the tissue, the distribution is extensive, because of which what happens is elimination will be a little prolonged. It takes more time for the drug molecule to come back to the systemic circulation and then enter into the excretory organ, that is kidney. So because of which you can expect the T of the drug to be prolonged. It happens, but it happens for a longer period of time. So etretanate is one of the drugs used for acne treatment, has unusually long elimination half-life of about 100 days due to extensive binding to body fat. It's just an example why the elimination of this etretanate is very much longer than 100 days. You can't even imagine 100 days elimination half-life we are talking. Half-life is 100 means full drug to get eliminated out of the body, you can imagine. You can just double that days. Okay. So etretanate, this is the reason, the justification why one of the reason may be the elimination is prolonged because it has having more affinity towards the body fat. And so maybe this other reason is it is highly lipophilic in nature. Because this drug molecule is highly lipophilic in nature, there is a chance that it has more affinity towards the body fat tissues, and because of which the elimination will be prolonged. So as overall, previously two class back, I have discussed about the factors affecting the drug distribution. There's one of the questions which you can expect, factors affecting the drug distribution. So hope you remember this, what we discussed. Tissue permeability of the drug, organ or the tissue size and perfusion rate, binding of the drug to the tissue components, and miscellaneous factors. Okay, and miscellaneous factors, I just highlighted about the age difference between the infant and the adult, also the condition like pregnancy, what happens. Because in every situation, there is a change with respect to the body constituent, which can affect the volume of distribution. Adding to that, okay, adding to that, one or two points I'm just highlighting. What are the overall factors that distribution changes due to? First one, change in blood flow, we know that because the rate of blood flow, Q is the one of the factor we have understood distribution half-life. To revise back in the distribution half-life, I told you the distribution half-life is dependent on the blood flow and also depends on the volume of the tissue and the R value. That is tissue to blood ratio, the concentration between the tissue and the blood ratio of that is called as R value. So change in the blood flow can affect. Why the blood flow can change? It can happen during the exercise. Okay, blood flow increases and the fraction of the drug reaching the receptor also increases. There's a chance that blood distribution will be fast. See, the volume of distribution, okay, is one of the mathematical way of understanding the distribution or a quantitative way of expressing the distribution. And that volume can be affected by all these factors. Okay, change in the blood flow also is one of the factors which can affect even that volume of distribution. Or I can correlate very well to the distribution time, that is a distribution half-life. Okay, so exercise can result in the high blood flow rate, so you can expect faster distribution to happen. Okay, so faster volume may not be much affected in this case, but the blood flow will be high, because of which what happens, the rate of distribution will be faster. So accumulation of the carbon dioxide in the tissue. So results in the pH of the certain tissues means due to any reasons, there's a chance that the condition, the environmental condition of the tissues can get affected, change. So there's a chance that carbon dioxide might get accumulated, and because of which the pH will change and may affect the level of drug reaching the tissue. So pH, we know that pH has greater effect. That actually governs the ionization and unionized fraction, which decides, the pH decides how much drug is getting ionized or how much drug is getting unionized. So this change in the pH will affect the change in the ionized and the unionized ratio, and also that can affect the volume of distribution. So that any condition which can affect the pH of the tissue can definitely change the distribution of the drug. And one of the reasons is accumulation of the carbon dioxide. Right now, I don't know what exactly this condition is called as, why the carbon dioxide accumulates in the tissue. There may be some pathological situation. I don't know much about this actually. So injury or the blood loss is another thing. Indirectly, blood circulation is affected because of the injury or the blood loss. The blood can rush to affected area and therefore affect the blood distribution during the injury. Okay, because of the too much blood is rushing towards that particular injured part. And the disease conditions, like especially the cardiac diseases. So cardiac heart failure decreases the cardiac output, resulting in the impaired blood flow. Okay. Same thing, meningitis can cause the inflammation of membrane of the spinal cord or the brain and increase the distribution to the brain. So like this, there are various conditions or the miscellaneous factors which can actually start affecting the distribution of the drug, which can affect overall the distribution. Okay, which might be, a lower distribution can become higher distribution. For example, in case of this meningitis, unnecessarily the drug is distributed to the brain, can result in the toxicity. So dosage adjustment has to be made knowing the condition of the person. So whatever the dose you would be treating for a person for managing the meningitis situation, you should very carefully plan the dose, knowingly that there is a disturbance in the barrier, that is a blood-brain barrier. Accordingly, the dose has to be reduced. Otherwise, unnecessarily the drug will rush towards the get accumulated in the brain because of easy permeation across the disturbed barrier. So like this, if we keep on discussing, there is n number of clinical situations we can start actually I mean, we'll get to know when we read the textbooks actually. We don't want so much in detail. So whichever is very important as an answer point, I'm just highlighting here. So when they ask the factors affecting the drug distribution, with the miscellaneous, you can add up this. Okay, you add up this points also as a part of the factors affecting the distribution. Fine. We'll begin with the next topic as a part of the distribution chapter. Is the drug protein binding. Got some idea already, like what happens when the drug binds to the protein? Definitely, it's going to affect the volume of distribution. When the volume of distribution is affected, elimination also is affected. It's a sequence. One thing is disturbed, rest of the thing you also start getting disturbed. So, but we are going to learn little in detail about the proteins which are present in our body. That is the plasma protein and the tissue proteins. Okay. So this is the situation, what happens to the drug molecule. Same thing, whatever I've been telling till now, after the drug binds to the protein in this box, when you see, there is a two possibility. The drug is present in the free form or the drug is bonded to the protein. Okay. So this drug, whichever is free drug, it can easily cross the membrane. But this drug bonded protein has a restriction. So at every places, there are the sequences or the situation happens so that the drug, whichever is bonded to the protein in the plasma protein, cannot easily cross, and the free drug can easily cross, and when it crosses, it can go into the liver, or it can go into the kidney, or even it can go to the drug target site for this therapeutic effect, the so-called as clinical responses. And here in the kidney, even the drug can get bonded to the carrier, and it can result in the active renal secretion. There is a process called active renal secretion. Or in the liver, it can bind to the enzyme and undergo metabolization. And these metabolites get excreted in the urine or through the bile. So like this, there are sequences happens to the drug after crossing the systemic circulation, and it comes out of the body by the excretion process. Okay. So fine. So this protein binding, like we know the definition of absorption, we know the definition of the distribution. Same way, there is a definition for the drug protein binding. So it's a complex formed by the drug and the protein, and which may be reversible or irreversible process. Okay, but most of the situation, it's a reversible thing which is happening in the body for any drug molecule. So this process of drug protein is reversible or irreversible. Irreversible happens because of the chemical interaction forming the strong covalent bond. Okay, so due to the chemical activation of the drug, which then attaches strongly to the protein or macromolecules by covalent chemical bonding, the results, because of which what happens, it's an irreversible. There is a chance that drug toxicity can prolong for the longer period of time. Okay, and there is, depending on how long it is binding, it can also exert the short-term toxicity also. The irreversible protein binding, though the word irreversible we are telling, it cannot be a permanent binding which happens with the protein, but it happens for the longer duration of the time. So this longer period results in the longer duration toxicity, or the shorter time. There is also possibility of the short time toxicity by forming the reactive chemical elements. There is a reaction happening. Chemical reaction can be happening, and that chemical reaction is mainly because of the covalent bonding. Okay, or by forming the some intermediate chemical intermediates also formed because of the interaction. But in the reversible process, at the worst is reversible. It is formed because of the weaker chemical bonds, and that happens because of hydrogen bonds or the van der Waals forces. Okay, so the irreversible, very rare event which occurs. Okay, though it's an irreversible we call, there is always that the drug will detach out of the protein, but it takes some long time. That's all. But that irreversible protein binding can happen because of the chemical activation process. Okay, and can also result in the formation of the reactive chemical intermediates. But in case of reversible protein binding, it's a weaker chemical bond happening with the hydrogen or the van der Waals process. So protein binding, drug protein binding is nothing but a complex bond between the drug and the protein, which is reversible or irreversible process. So coming to specifically the plasma protein binding or the plasma proteins, these are the major plasma proteins which are present in the blood. So as I told, blood has many components and plasma is the fluid component of that. And there are even the proteins. Okay. And there are even antibodies which are protein in nature. Okay. So we are talking about the proteins which are present in the plasma. Okay. They are the albumin, alpha acid proteins, glycoproteins, okay, and the transcotin, and lipoproteins and the transcoatins. So these are some of the examples of the proteins. I don't expect you to by heart all these numerical values, but just for to know that which is in the higher percentage. Albumin compared to rest of them, albumin percentage is high in the blood compared to any other proteins which are present in the blood. So we'll go in detail, not detail, very briefly about the individual proteins, because we need to know about the protein to get idea whether the drug can bind to that particular protein or not. And because drug protein binding is structural specific. You cannot expect all the drug molecule to just come and bind to whichever protein which are present in the plasma. They are structural specific. It depends on the type of the drug molecule and its specificity towards that particular protein. So all this, so it's a structural specific event which is happening. The drug protein binding is a structural specific event. So albumin, which is present in the higher percentage compared to the rest of the protein, its concentration is approximately between 3.5% to 5%. And these are present in the plasma, extracellular fluid, that is interstitial space, skin, and the muscles. So these proteins are not only limited to the blood, they are also present in the extracellular fluid, skin, and the muscles, and also in some of the other tissues. But more it is present in plasma. So that's what we are supposed to remember. And its concentration in the interstitial fluid is 60% of that of the plasma. Initially, it is very much the higher percentage is present in the plasma. Next comes the interstitial space. But that interstitial space, whatever amount is present, that is 60% of the plasma protein. If the plasma protein is 100% or 100 microliter, then the interstitial fluid will be 60, depending on the gram per liter. Okay, it is a gram per liter. We express the unit, its concentration. If it is 100, then it will be 60, but it will be much higher in the plasma. So one of the function which you can add up to the albumin is it maintains the osmotic pressure of the blood. Okay, and also it acts as a transporter of the endogenous and exogenous substances. Okay, it helps in movement of the some of the substrate molecule, endogenous substrate or exogenous substrate. Endogenous it comes inside, exogenous it will be sent outside. It complexes with the fatty acids, bilirubin, cortisone. These are all the common components which you expect to be present as in our body, other than the drug molecule. So, if some important functions are, it manages so it balances the osmotic pressure, it transports the endogenous substances, or the substances either by endogenous process or exogenous process. Coming to specific, when we talk about the protein binding, it binds to weak acids, that is anionic drugs. This you have to memorize by electrostatic and hydrophobic bonds. Examples are salicylates, phenylbutazone, and penicillins. Okay, so it has more affinity towards the albumin. Has more relatively, it is having more affinity. The drug molecules which are weak acid in nature, okay, which is anionic, they have more affinity towards the albumin. Okay, and there are certain binding sites which are present in it. When I say binding happens to form the complex, there are binding sites which are present in the protein, and these binding sites may be like many types in the protein, which are present more than one in number also. Okay, so one of the important binding sites has to binding human serum albumin. It is called as human serum albumin 2. HAS2 it is named as, is one of the protein binding site which is present in the albumin. with high association constant of 104 to 104. This is one of the numerical value, okay, quantitative value, which gives us idea about the binding capacity of that drug molecule. That means how much they have liking to each other. That's called as association. Association is combining. Dissociation is getting detached. So HA has two main binding site, has higher association constant, okay, of 104 to 106 molar, okay, per molar. So one of the value you actually the unit used for expressing the molar constant. And the sites are charged positively and bind mainly to the ion. That's what I told you here. It is more of affinity towards the anionic drug molecule, and with the binding constant of 104 to 106 molar unit per molar unit. Okay, and some of the drug that is anionic in nature or non-steroidal anti-inflammatory drugs can be some of the drugs which you can expect to have the greater affinity towards the has to binding site. There are other sites also called as site 1 and the site 2. Site 1 is specifically for the phenol. I told there are varieties of the binding site present in the protein, which may be more than one in number, okay, and there are many types also. So site 1, there are some of the very unique, specific one I'm just stressing upon. Site 1 is for the phenylbutazone, sulfonamide, phenytoin, and the valproic acid. Okay, there's a mistake here, delete of this AD. The site 2 is specifically for the penicillin, propionic acid, medium chain fatty acids, and benzodiazepines. So these are some of the examples of the drug. There are some sites called as site 1 and the binding site called as site 1 and the site 2. And also there's some site called as oxycan. Both sites for the called as warfarin site, that is the warfarin can bind to even this oxycan sites. So I want you to just remember that albumin is present in the plasma and other parts of the body. They're present in the higher concentration, okay, and it has more affinity towards the weak acidic drug molecules and has some different binding sites called as a has 2 as the main binding sites, and there are even other sites called as the site 1 and the site 2. See, when we go in detail, there's a lot of things to understand about this binding sites. Since it's not required, and also I'm not very much good in this aspect.